Polyethylene terephthalate, or PET, is one of the most widely used plastics worldwide and is linked to 8.8 metric tons of carbon dioxide emissions per year just in North America alone. PETase, the plastic degrading enzyme produced by the recently discovered Legionella bacteria, represents an exciting bioactive approach to eliminating waste. A novel approach is to use alternative PET copolymers with lower crystallinity, facilitating their breakdown by PETase. In this case, I studied the enzymatic degradation of polyethylene terephthalate glycol, or PETG, as a valuable alternative to PET in the future, as well as a current target for PET to be recycled into. The main distinguishing feature of PETG is the ability to achieve similar hardening levels to regular PET at a much lower level of crystallinity. Given that the activity of PETase diminishes as the crystallinity of PET increases, the lower crystallinity of PETG could significantly enhance its susceptibility to enzymatic degradation. To answer this question, I first performed transformation via electroporation in order to express plasmid containing PETase gene in E. coli K12. The transformation was then validated using selective media containing ampicillin and plasmid recovery. Additionally, plasmid samples were evaluated using PCR and restriction digestion analysis pre and post transformation. Next, I induced the production of PETase by producing IPTG to the transformed E. coli K12 culture for 18 hours. This was followed by enzyme extraction using centrifugation, a lysis buffer, and sonication. The next process was his-tag purification via copper affinity chromatography, which was performed due to the presence of the histidine tag on the enzyme. I then used a size exclusion column to complete a buffer exchange of the his-tag purified PETase sample. To visualize the PETase extracted, I used SDS page analysis with the samples of each stage of the enzyme collection process. A further IPTG induction validation was also conducted by performing the PETase collection method on two transformed cultures, one induced with IPTG and the other was not. The enzyme samples were collected and further evaluated using nanodrop analysis. I then treated PET and PETG plastics in addition to solutions containing bez-hydroxyl-ethyl terephthalate, or PET, with the extracted PETase over a period of 12 days. After which, I used UPLC analysis on samples in order to quantify the degree of enzymatic degradation. The plasmid size, PETase gene size, and expression of ampicillin resistance matched the characteristics expected from the PET21B plus PETase plasmid. This was true for plasmid samples pre and post E. coli transformation, thus confirming a successful transformation. SDS page analysis confirmed that not only PETase was successfully extracted using the methods of the study, but also it was able to purify the samples at each stage of the process. Additionally, nanodrop analysis indicated that the enzyme concentration from the induced culture was over 2 times higher than the non-induced culture. This does confirm the effectiveness of IPTG induction in this study. After evaluating UPLC analysis, it was clear that no detectable concentration of PHET was found in any of the PET or PETG samples. This, coupled with the lack of mass loss or visible degradation in any of the treated plastic samples, indicated the lack of PETase activity. Furthermore, unexpectedly, the treatment of solution containing PHET did not produce the expected results of decreasing the overall concentration of PHET in the solution. Overall, the lack of degradation of PET, PETG, or PHET warrants a further investigation and highlighted the limitations of this study. However, this study did demonstrate a successful method of collecting a purified PETase sample from transformed E. coli K12, which still suggests the potential of viability of using a bioactive approach to the plastic pollution problem.